Chapter 5, Viruses and their multiplication. Section 1, what is a virus? Now, a virus, well, viruses just aren't anything. As far as, living organisms are considered, they're not living cells. They don't have, cytoplasmic membranes, cytoplasm. They don't have enzymes. They don't have a metabolism. Everything that, we use as our check boxes to say, yes, this is a living cell. This is a living thing. Yeah, viruses just don't. It's why you won't see them listed in any tree of life things, because they're not. They are obligate intracellular parasites. If they're not in a host cell, and notice I said host cell, not a host. You can be a host for bacteria, for parasites. For a virus host is one of your cells, a specific cell type in you. So when I say host, I'm talking cellularly, not you as the organism. Well, it needs a host cell, this obligate intracellular parasite. Outside of a host cell, it is about as living metabolically as this cup. Nothing going on there metabolically. Same with a virus. Now, once the virus gets inside a host cell, then things start to happen. The virus genome, the virus proteins, will hijack the host cell, turn that host cell into a virus-making machine, hence the parasite portion. Ultimately, the host cell will die because of this, but not before it makes hundreds, if not thousands, of virus copies. So this is what they refer to as a viral infection. The virus getting into the host cell. So just because you get a virus inside you doesn't mean you're infected. The virus has to not only get inside you, but has to get inside one specific cell type inside of you. Then you have an infection. And the viruses, they come in all shapes and sizes. Here you have the Zika virus to the far left. You have, the mosaic virus, the tomatozoic virus. It's got this helical pattern going on. You've got this weird shaped, amoeba virus, and so on and so forth. Just different shapes, different host cells that they will infect. Now, when it comes to the virus, there's a few things of nomenclature we have to get out of the way. All right, make sure we're all on the same page. The virus has what is referred to as a capsid. The capsid is the part of the virus that houses, surrounds the genome. Okay, it's a protective protein coating. Now, that capsid may or may not be wrapped in a membrane. I know I just said viruses don't have cytoplasmic membranes, but that's not what this is for. It's not carrying in cytoplasm. It's just another protective layer. If the virus has this protective membrane, it's referred to as being enveloped. If the virus does not have this protective membrane, it's referred to as being naked. And yes, there are so many jokes I could make right now, but I'm not, because I am trying to be a mature adult. So when you have the capsid and the nucleic acid together, you have what is referred to as a nucleocapsid. In a naked virus, that's all they are, a nucleocapsid. But the envelope virus is this protective membrane, this phospholipid bilayer, which generally is going to be taken from the host cell cytoplasm. We'll talk more about that later. The lipid bilayer wraps around the nucleocapsid, another layer of protection. Whether it's naked or enveloped, there's going to be surface proteins. These surface proteins are what allow these viruses to interact with our cells and force their uptake. Side note, if you've been following any of the talking heads in the news for the past year and a half, that whole COVID pandemic thing going on, you've heard them talking about S proteins, the viral spike proteins. That's what they've been referring to. These proteins on the surface that allow for the virus to gain entry into our cells. If you've had your shots, you've got the vaccines. The vaccines are messenger RNA, or the instructions in the messenger RNA form, so that these viral spike proteins can be made by our own cells and then exposed to our immune systems. Naked, enveloped. The nucleocapsid is the genome and the protein. Genome and the protein capsid. Now, what the artist didn't draw here, and I've got a bit of a problem with that, is that there should be spike proteins coming out of this too. Because this is also going to need the ability to bind to cells. So these both serve the same purpose. Viral proteins that interact with receptors on our surface of our cells that will force the uptake of either the naked virus or the enveloped virus. Now, do not mistake just because, oh, look, here's the nucleocapsid, and this nucleocapsid looks exactly like that nucleocapsid, so the virus can be enveloped or naked. No. That strain of virus, influenza, COVID, poliovirus, monkeypox virus, whatever. All polio viruses are going to be naked. All influenza viruses will be enveloped. They do not switch back and forth. Okay, millions of years of evolution, they're either going to be naked or that virus will be enveloped. Okay, it's not a mix or match thing. Here's a little video you can watch. I'm not going to sit there and play it now, but it's in the handout, so you can go to the handouts and hit play. We talk about viral infections. We will sit there and say that it's a virulent infection because the virus gets into one of your cells, one goes in, hundreds, if not thousands, come out. And as they're coming out, they're destroying the host cell. They're causing tissue damage. They burst out of one and then diffuse to the neighbors. One goes in, hundreds, thousands come out, burst those, and spread that way, doing damage. Virulent, also known as lytic. Can also end up with what is referred to as lysogenic. It's where the virus goes and infects the cell and then hides. The viral genome just inserts into our chromosomes. HIV does this. Herpes does this. You get infected, one of your cells gets infected with the herpes or the HIV virus. The chromosome, the DNA genome, or a copy of the genome, of the viral genome that's now in DNA form. I'll tell you why I'm being a little funky on that in a few lectures from now. Will insert into our chromosomes, randomly insert someplace, and that's where it'll hide. That's why herpes, whether it's herpes, human herpes simplex 1, human herpes simplex 2, once you get it, you've got it for life. Because because it inserts into our chromosomes of our stem cell lines that make up, certain cells in our bodies, or it inserts into the neurons. HIV does the same thing, but it inserts into CD4 T cells and into the germline for the CD4 T cells. So that's now part of the chromosome of CD4 cells. Turns out to be the same cells that are responsible to helping kill viral infected cells, is where it hides. Cool. Now, why on the last slide I was a little bit flaky when I was going, oh, the genome, the DNA, the copy of, the genome does the DNA, blah. Turns out viruses break rules. Not only are they not living, they don't use DNA all the time, or their genome. We, living things on this planet, and pretty much all living things, prokaryote, bacteria, archaea, and us eukaryotes, plants, fungi, protists, animals, we all use DNA as our genomes. RNA is the transient thing. Viruses, not so much. Certain viruses are going to use DNA. Certain viruses are going to use RNA for their genome. Of the DNA viruses, some use the double-stranded, double helix, double-stranded DNA, as you've come to know and love. Other DNA viruses have single-strand DNA. Same with the RNA viruses. Their genomes are made of RNA. Could be single-strand RNA, could be double-stranded RNA. The only thing that we do know is that their genomes are going to be quite small. Our genomes measured in the billions, B. Bacterial, archaea genomes measured in the millions, M. Viral genome measured in the thousands. Every living thing on this planet has at least one virus that will infect it. All bacteria, archaea, animal, plant, protozoa. Supposedly there are viruses that infect viruses. Not sure how that works, but I think part of it has to be that they have to infect the same host cell at the same time. And then, virus B hijacks the viral A hijacked cell and overrides everything virus A has done. We spend a lot of time spending, and money studying the disease-causing viruses. Just because, well, for one, it's interesting. Two, if we can figure out how they work, we can try to figure out ways to cure, if not treat, virus infections. But not all virus infections are damaging, deadly. You look at the plant here, Arabidopsis. The plant to the left is not viral infected. Yes, remember, even plants get viruses. The one on the right has. Under nice and water is in plenty, this condition. Turns out the virus-infected plant, it's not growing as well as the not infected. But when they go through and they cut off the water, and they now put it under drought conditions, look who's surviving better now. Something about the virus infecting the plant cells makes them more resistant to water loss, to the slow death due to drought that you see here with the non-infected. Got to remember, these viruses have been around longer than we have. So as the new species has evolved over the past billion years, us animals, us plants, us eukaryotes, well, the viruses have evolved right alongside of us. So it's been a co-evolutionary event. You bounce back to look at the last slide. you'll see that, it mentions there are certain, insects that the path of their life cycle will be determined whether or not, due to whether or not that organism, that individual organism, has been infected with a virus. If not, it grows a certain way. If it has been infected with a virus, well, it gets smaller and now has wings and is more mobile, is able to fly great distances.